Good morning, everyone. Today we are going to discuss about the important 4 marks questions from cell cycle and cell division. So now first question is interface does not involve rest comment. So this is very important question in the IP examination, right? So interface. So here cell cycle. So cell cycle and cell division, here mainly two phases are involved in the cell division, that is interphase and M phase. So here interphase starts with the G1 and ends with the G2 and followed by the that is mitotic division, that is M phase. So here cell lasts 95%. That means here the cell remains 95% in interphase only. So cell lasts for 95% in the interphase only. So here that is, if you take human cell, approximate cell division time of a human cell is 24 hours. Approximate cell division time is 24 hours in humans. In humans. So here cell lasts for 95%. That means approximately 23 hours cell remains in approximately 23 hours cell remains in interphase only. Accurately 22.8 hours accurately. So cell remains in interphase. That means cell involved in resting phase. So here the question is, interphase does not involve rest comment. Though it is redundantly described as resting phase, it does not involve rest. So now let us discuss about the what are all the different phases of interphase, right? So in the interphase, so there are three phases are there, that is G1, S, G2. So G1 lasts for 11 hours, whereas G2 8 hours, and whereas S 8 hours and G2 4 hours. That is, so 11 plus 8 19 plus 4 23 hours. So cell lasts for 23 hours in the interphase. So here in the G1 phase, what happens? So cell is metabolically active. Cell is metabolically active. Metabolically active, and synthesis of proteins takes place. So synthesis of RNA and some proteins occur here in the G1 phase. So G indicates the gap 1. G indicates gap, right? And here, so cell is metabolically active, and synthesis of RNA and proteins takes place, and most of the cell organelles duplicates here in the G1 phase. So most of the cell organelles duplicates here in the G1 phase. And next, G come to the S phase. So the cell enter into the S phase after the completion of G1. So in the S phase is also called as synthesis phase. Synthesis phase. S phase is also called as synthesis phase. So here DNA duplication or replication takes place. So duplication of DNA or replication of DNA takes place in the S phase, and centrioles duplication also takes place in the S phase. So centrioles duplication takes place in the S phase. So here in the S phase, the amount of DNA is increased. The amount of, that is very very important, amount of DNA is increased, but the ploidy never changed. Ploidy never changed. For example, if you take cell in G1, cell in G1, so the cell in G1 is a diploid condition. Diploid condition. For example, so this is the chromosome or a cell which is diploid in condition, and here the ploidy in the S phase also same. The ploidy in the S phase is also 2n. S phase is also 2n. But here the thing is, the amount of DNA gets duplicated. The amount of DNA gets duplicated. That is increased. Amount of DNA is increased. For example, the amount of DNA in G1 phase is amount of DNA in G1 phase is 2C, 2 concentration. So whereas in S phase, the amount of DNA becomes double, 4 concentration. So the DNA amount initially in the G1 phase is 2 concentration, whereas in the S phase it will the amount of DNA increased, the amount of the concentration of DNA increased. So this is about the S phase. So duplication of DNA or replication of DNA takes place in this phase, and centrioles duplication takes place, and amount of DNA is increased. That means it may get double. But remember, ploidy never changed. That is, in the G1 phase, if the cell is in 2n condition, here in the S phase is also the cell is a 2n condition, diploid condition. But remember, only amount of DNA, that is concentration of DNA, gets increased in this phase. Next, G2 phase. So events which occur in G1 phase are also occur in G2. That is, cell is metabolically active. Cell is metabolically active, and synthesis of RNA, proteins takes place, and here chloroplast and mitochondria mainly duplicated in this G2 phase. Chloroplast and mitochondria mainly duplicated in this phase, right? So in the G2 phase, almost all the events so which occur in G1 also occur in G2 phase. That is metabolically active. The cell is metabolically active, and synthesis of RNA proteins takes place, and cell organelles duplicate. That means especially chloroplast and mitochondria. Chloroplast and mitochondria duplicates here. Right. Next, after completion of G2, that is, it is a last phase of interphase. It is a last phase of interphase. In the interphase, G1, S, G2. And if you take whole cell cycle, the sequence should be start from G1, S, G2, M. So remember that. So G1 sequence is very important for Neet point of view. G1, S, G2 and M. So here up to G2 phase, it is considered as interphase. So up to G2 it is interphase, right? So cell remain 95% in the interphase only. That means cell is in, usually it is called as resting phase, but it does not involve rest. It involves in various activities like synthesis of RNA and protein, cell organelle duplication, synthesis of DNA, and synthesis of other organelles like chloroplast and mitochondria takes place in the G2. And then it enter into the M phase. After completion of interphase, it enter into the M phase. What happens in the M phase? So in the M phase, first karyokinesis takes place. That is division of nucleus. Division of nucleus, and followed by cytokinesis. And then so two daughter cells will be formed. And if the cell does not want to enter into the cell cycle, the cell enter into the G0. So this G0 is also called as quiescent stage. Quiescent stage. So heart cells usually do not divide. So if the cell want to divide, the cells enter into the G1 and the cycle repeats. And the cycle repeats like this. So G1 yes, G2 M. So remember, it is very important question for the IP point of view. Next, let us discuss about another question, that is key features of meiosis. Key features of meiosis. So here, what is meiosis? What is meiosis? So meiosis is a reductional division. Meiosis is a reductional division. Meiosis is a reductional division. That means number of chromosomes are reduced to half. For example, so this is a parental cell, right? It undergoes meiosis division. So first, initially, meiosis involves mainly meiosis 1 and meiosis 2. So in the meiosis 1, the actual reduction division takes place. The actual reduction division takes place. Dyad of cells will be formed. And meiosis 2, it results in the formation of 4 haploid daughter cells, which all are different. So haploid N, N, N. So meiosis 2. So this meiosis 2 is equivalent to the mitosis. Meiosis 2 is equivalent to the mitosis, equational division, because in the mitosis what happens? Equational division takes place. So it is parental cell which is diploid in condition. It undergoes mitosis division to form 2 daughter cells. So the ploidy levels remain unchanged. So parental cell 2n, daughter cells also 2n, just like in the meiosis 2. So here it remains haploid only, but the actual reduction division takes place in the meiosis 1. So actual reduction division takes place in the meiosis 1. So meiosis involves meiosis 1 and meiosis 2. So meiosis 1 and meiosis 2. So meiosis 1 is also called as a actual reductional division. In the meiosis 2, what happens? In the meiosis 2, equational division takes place. So meiosis involves meiosis 1 and meiosis 2. So meiosis involves meiosis 1 and meiosis 2. meiosis 1 and meiosis 2. So in the meiosis 1, so we have different stages, that is prophase 1. So this prophase 1 is very complex and it has different substages. So prophase 1 have different substages, that is leptotin, zygotin, pocketin, diplotin, diakinesis. So these are the 5 substages of meiosis. So prophase 1 of meiosis 1. So after prophase 1, metaphase 1 and anaphase 1, and telophase 1, So after the end of the telophase 1, so the cell enter into the meiosis 2, So here the gap or the phase between the telophase 1 and meiosis 1 is called as interkinesis. So here meiosis 2 involves prophase 2, metaphase 2 and anaphase 2 and telophase 2, right. So here no need to write this in the examination. So we shall move to the previous slide, right. So here the question is the key features of meiosis. So here it involves 2 sequential cycles of nuclear and that is cell division, nuclear and cell division, and but only single cycle of DNA replication takes place, but only single cycle of DNA replication takes place. So 2 sequential cycles of nuclear and cell division takes place in the meiosis, very important. So 2 sequential cycles of nuclear and cell division, but only a single cycle of DNA replication takes place, very important. So this statement is very important for objective point of view also, right. So meiosis 1 initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. So meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatids at S phase. So meiosis involves pairing of homologous chromosomes. This we are going to discuss in the prophase 1 of meiosis 1. So involves pairing of homologous chromosomes and recombination between them. So this point is also we are going to discuss in the prophase 1 of meiosis 1. And after the end of the meiosis division, what happens? 4 haploid cells are formed at the end of the meiosis, and all are different. They are not same. All are different. Right? And meiosis occur mainly in the germ cells or reproductive cells. So germ cells are reproductive cells.